Uh, Good afternoon, ladies and gentlemen. It is my pleasure to introduce our next speaker, Dr. Tomasz Kolanowski. I know him for over a decade, so I witnessed his master's degree graduation in biotechnology, Poznań University of Life Science, gaining the bachelor in bioinformatics at Adam Mickiewicz University, and Ph.D. defenses in our institute, which he completed with honors. Dr. Kolanowski received multiple awards, including FNP START and Kulczyk Foundation fellowships. He was involved in several grants, also as principal investigator. His scientific interest started from stem cell therapy of infarcted heart, then he moved towards modeling heart diseases with stem cells, but since he came back from Germany, where he did his postdoc fellowship, he became an expert in tissue engineering. And his expertise started to grow. During the collaboration with the Famicor Therapeutics, he was given the challenging project regarding implementation the manufacturing of first Polish CAR-T therapy. And I must say, he did it almost from the scratch. And there he is, leading the R&D department with over 20 people and being the member of Famicor board as a chief scientific officer. I'm sure that you will enjoy and appreciate his talk as much as I appreciate and enjoy working with him together. So, Tomek, the floor is yours.
First of all, thank you for this very nice invitation. I didn't expect that. I hope the presentation will be at least the same interesting and the same enjoyable for you. Today, I would like to discuss about CAR-Ts, as we are like in a Hematology day right now, talking about the CAR-T implementation. But I would like to tell you a bit more from the scientific point of view, from the scientist's perspective. I have, as Natalia mentioned, I have conflict of interest here as well. So my presentation would be about the initial view on a CAR, what it is, what it's about. Then I would like to go through the mechanistic approach to the engineering of these constructs, talk a bit about the cell sources and what does it mean for us and for the clinics and for efficiency of the therapies. What are the hurdles and perspectives? Clinical state of the art, because we are in a hematology day, so we need to know what is going on. And then manufacturing issues and some process organization, which is not easy, as you may recall. So the CAR-Ts have been firstly described in 1989. I was 3 at the time. So this was 30 years before they came here to FDA approval. And it was completely new generation, like second generation CAR-T. And this is an example of ex vivo gene therapy, actually. So the CAR, so chimeric antigen receptor, it's a receptor that combines different modules, different subunits from different proteins, mostly available in our body. First part is like this extracellular antigen binding domain, which is normally kind of an antibody. Then through the transmembrane domain and intracellular co-stimulatory and stimulatory domains, often recalled as 4-1BB or CD28, and crucial one for this intracellular signaling, CD3 zeta, which is coming from the TCR. This CAR allows us to target the antigen that we would like to have. This is 319 as a most common one. But then what it does, it activates the T cell in this case, and to make this cytotoxic activity. About these generations I mentioned before. So first generation CAR was not active at all. I mean, it's all about the internal stimulatory domains, and with the first generation, we had only CD3 zeta. It activated the cell, however, the cells were not active clinically and couldn't maintain this cytotoxic activity. Actually, with the second generation, it's a generation that actually really reaped the success of the CAR. So this means that actually, apart from CD3 zeta, so T cell activation domain, we have CD28 or 4-1BB activatory domains, and this really makes the card working. There are third generation cards that combines all these subdomains. However, they are not so much popular as we expected. And the fourth generation of a car, which is not here on the screen, it's only at the 0.6 here, is about armoring. So adding something on to the CAR T to make the cells really doing something else or being better than they are normal, than only with the CAR. There are like similarities with the TCR, so the CD3 active mechanisms of action and CARs. However, they do differ a bit. Although CAR comes from the TCR idea, the TCR has three really distinct signals within these activations. So activation of the cells through the TCR need to come from the TCR itself, together with the CD4 or CD8. Then you need to have additional signal number 2 from 4-1BB or OX40, and then interleukin 2 receptor through JAK signaling need to be added as well. In terms of CAR, it's less discrete. So the activation goes it's less controlled. However, CAR is less potent. So this means that actually when you activate the cell, T cell, through the T cell receptor, actually you have items which are like motifs in the TCR receptor that activates the subsequent signaling. And these items in TCR, you can observe 10 of them. In terms of CAR, it's between 3 to 6. So it means that actually this pyramid of activation is less potent at the very beginning. What are the differences between TCR and CAR? TCR forms very well-organized, demarcated immunological synapses. There are like 3 supramolecular activation complexes, and you can see like the central one, the spherical one, and the distal one. Why this is an important part? Because actually this is all about the steric interactions between the cells. You can see that TCR complex is in a central supramolecular activation complex, and this together with the CD4 and CD8, they are closely binded together. In terms of farther, you have the peripheral SMAC actually, with the myosin and actins within the cell regulating the spacing between the cell membranes. And then there is a glycocalyx part, which is the distal part. In terms of the why is this important, you can see what are the differences, like the physical differences between the proteins. TCR complex is about 7.5 nanometer from this cell membrane, whereas CD45 is like 50. So you cannot reach the receptors and binders between the cells actually if you don't have the cell membrane well organized. In terms of CAR, this is much less organized structure. So CAR just reaches the target antigen, and around the CAR there is like kind of a microcluster formed and organized. And then what you have around is additional domains, and these microclusters just appear spontaneously. This means actually this less organization of the CAR leads to more rapid degranulation of the cell, so activation and degranulation of the cell, but the same disengagement from the target cells. This actually means that you can gain faster killing. So what you have between the TCR and CAR is less organized and less controlled, but it's faster for killing. Okay. So this is a classical CAR construct design. You can see there are different possibilities to design a different way. Professor Gill, with her great talk before, has mentioned about different possibilities and different CAR constructs. You can have switchers, and you can have different antibodies. Starting from the binding domains, this is not only antibodies that can be used. There are ligands, cytokines, nanobodies you can put on. Then you have hinge, transmembrane domain, co-stimulatory and activation domains, and this armoring, as I mentioned before. And I would like to right now go through each of these parts with you to show you what you can do with the CAR actually and how you can prepare this for action in the cell actually. First of all, these binding domains. So binding domains has a multiple possibilities. First of all, you start with multimodality of this binding domain, so you can combine different binding domains together, right? You can have a tandem CAR, which is having like two domains on the same CAR particle. Then you have dual CARs, which are basically two different CARs on the same cell, and then you have multi-specific, just a mixture of CAR, different approaches. Then you have these bites, which are like double binding kind of adaptor antibodies. And then you can have adaptor molecules, which allows you to use different kind of antibodies and somehow to functionalize your CAR to the different targets. Then you, by designing a binding domain, you can increase the specificity of the CAR, which is I will tell later on, which is sometimes tricky to reach. So you can increase specificity through dispersion of the activation signal. And you can see here, like you can have like these activation signals in two separate CARs. Then you have need to be both of them need to be activated to get the signal an opposite way. This is a B one. And then you can use natural proteins that actually are in the cell, so TCRs and interleukin receptors you can use as a co-stimulatory for your CAR. Finally, that's a kind of an interesting part. You can use logic gating. This means that actually your CAR can appear only when there are some particle environmental issues and features going on. So, for example, if you would like your CAR to be only expressed in low oxygen conditions, so then you have like hypoxia responsive elements, HIF, and this factor controlled by this CAR is controlled by this factor basically, and then it appears only in these kind of cells. There is like an example of the Notch receptor activation, and through this activation, then you get the gene turned on, and then the signal occurs. So this is again kind of a logic gating. Example of the binding domain engineering is like AVID CARs that are published like 2 years ago. Actually, the principle was that Salter Benjamin, they engineered this antibody they have. They have really nice high affinity antibody. They engineered by introducing mutations these antibodies to get into the low affinity antibody. You can see on the C. And then with this low affinity, they reached the moment when the singular antibody were not able to lyse the cell specifically. However, the tandem one can do it. So it means that actually it's like CAR is turning on only when it's becoming in a complex. This complex can be then used for different applications as an on switch, for example. So you use the dimerization factor, dimerizer, and you can use it as an on switch. You can use it as an end switch in terms of the different targets that you have. And these targets could be on the surface and could be soluble antigens as well. So this is quite a nice example of the engineering of the binding domain. Then the hinge. If we're thinking about the hinge at the very initial stage, hinge was really neglected because what it was all about, it should provide the flexibility just for your binding domain to get this, to get rid of this, overcome this steric hindrance that can happen. However, we know now that the spacers, so this part of the hinge regions, are very necessary when you want to target the proximal membrane proximal domain. So you want to reach out with your CAR binding to this proximal antigen. This means that actually, if you engineer this hinge, this spacer, and shorten it down, you can see that actually from blue to purple one, you can see down activation, and activation is going down with the cell because it's within the cell expressing CAR, because it cannot reach basically this cell membrane and proximal domains of the target antigen. So that's a very crucial one. What you need to think about is that even if you use the big spacer, like very large one, here it's IgG4, sorry, spacer, what you can get, although you can reach very nice effects in the C, you can see like in vitro assays, so basically long and short one are actively destroying the cells. You cannot reach it on the mice level, so you can see that actually within the long CAR, you cannot observe this diminishing of the tumor burden. This is because it's blocked through the like different receptors. In this case, it's a Fc receptor on a different cells, and actually it cannot be working properly later on. So what to do with this part? Because you need to reach this. So how to do it? What to make with it? This hinge engineering can optimize CAR in different ways. Here you can see in A that PSCA, it's just an antigen. P1 CAR, this is spacer, a very long spacer from IgG1 derived with CH2, CH3 subdomains. And you can see that this is exactly what it's doing. It's not active within the in vivo. But what you can do, you can mutate this one and regain the action of the CAR in vivo. And these mutated parts can be active. However, what is then happening, you can have an issue with the tonic signaling. So if you have really long subdomain, which is going around there and hanging, it means that it's always constantly, even not binding the antigen, it's always constantly activating the CAR, activating your cell. And this is what is seen in here. All of these CARs actually activate the cells, and they're constantly proliferating because they are active, so they are prone to exhaustion. How to avoid that? You can engineer to cut it off, right? So that's the solution, and you can see after cutting it off, you still have like the cells which are not active, like standardly active. However, obviously, then if you cut it off, you cannot reach the antigen again. So then, what they did in this publication here, they used only one of the parts of the like shorter spacer region, and they really regained this function, although not being like overactive in a way. Now, what you can see here in PSCA dim population of the cells. So when you don't have too much antigen, you actually, when you have really short CAR, this X2 CAR in a green one, you don't see an action in them. However, when you have the CAR which is engineered to be a bit longer, then the action is regained basically because you can still reach it. And this is something that is shown in here. That's the summary. So you can see that even with this such a simple thing as a hinge region, you really need to be very careful to engineer it properly to get it into the target you want, right? Because if the target is very proximal to the domain, you need to have an appropriate length of this. And this shouldn't be disturbed by any other factor. Then the transmembrane domain. It seems quite simple, right? It's just anchoring to the cell membrane. However, transmembrane domain can influence many factors of your CAR. First of all, it's stability. It's known that actually stability of the CD28 domain is larger and it's bigger than CD3 zeta. It's because CD3 zeta then can mediate actually CAR dimerization into TCRs. So it means that actually, although it's not stable, then it can gives you additional boost through the TCR action. And that's the molecular interaction that you can get into with just this transmembrane domain. What is even more important is that CD8 versus CD28, you can see the differences in the susceptibility of your cell expressing this CAR with the activation induced cell death. So there are different functions that you can observe between different transmembrane domains that can influence your CAR. Right. So then the intracellular signaling domains, so these famous ones. With the famous ones, we all know that CD3 zeta, it's a classical domain that consists of these item motifs, which is activating the CAR. Then you need this CD28, as I mentioned before, and 41BB. These both domains provides you a proper activation of the CAR. And that was it basically for the long time. There were several domains that were tested as well, but they function in a different way. However, this month, actually, November 2022, sorry, a pool CAR approach actually was proposed for the discovery of the new domains actually that can work better or work similarly. And Goodman and all, they provide this kind of a pool approach, meaning they use a classical CAR, CD19 CAR, but they exchange this co-stimulatory domain. And you can see in these brackets there that they're pooled a library of these signaling domains from the different cells, shown in B, that they came from T cells, NK cells, DC cells, granulocytes, different ones, and then just put it in to the bunch of the T cells, right? So they create a pool of the CAR, and then these CARs were repeatedly activated by the CD19 plus cells, right? And this is D, you can observe. They add every day new and new ones. And then what they want to show and they want to screen is that how the cells behave afterwards. And what they propose and what they show is that there is a bunch of this greenish part called activators, would say, stimulatory domains. And you can see within this group, you can see 41BB obviously and CD28. However, there are a few more. This TACI, so TACI or TACI, and BAF. These two domains were proposed as new domains for the activatory domains that could be used within the CARs. What is interesting, what they observe, that there are some domains that actually act in a different way. They inhibit CARs. So KLRG1 domain was discovered as a quite potent inhibitory domain. That's quite interesting when you think about this logic gating and about controlling regulation of the CAR. What is even more important, what they observed is only 41BB domain. Actually, you can see it in C. When you look at this 41BB, you can see that only this domain could, after 24 days, so prolonged time, could promote CD4 cells, so helper cells. This means that this domain tends your cells to be more active over time, so to be pronounced within the body, within the in vivo experiments, or in vitro in this case, to be there. What they did next, they used this quite fancy technique of single-cell RNA-seq and site-seq, so combine basically single-cell RNA experiments with the secretome experiments, so they can really organize the subpopulations of the CD4 and CD8 cells that were defined by CAR. What is out from there? What you can see in this lower part, that different subdomains actually activate cells in a different way. What is out of it is that actually, again, here you can see the stimulated CD4 and CD8 cells and grouped through the groups, clusters of the cells that are more activated ones, you can see in the reddish color, and more less activated, more naive one. And you can see that actually from the top, untranslated, untransduced cells, obviously they are not so active. Then you have this inhibitory domain, which is, although CAR itself is activated, it's not activating the cell, so it's really inhibitory in this part. Then you have CD3 zeta alone, and then additional subdomains and molecules, and you can see that this both TACI and BAF receptor, 41BB and CD28, there are really potent activators in here. This was confirmed in mice, so in vivo experiments, and you can see that on this green part, you can see that this is really potent. These all 4 domains, 4-1BB, CD28, and BAF and TAXI, they are all potent activators of the CAR therapy in vivo. Whereas the KLRG1 is not acting, actually the effects are the same as a non-transduced cells. What is quite interesting is that what they observe is when you use the CAR with the BAF, so this new costimulatory domain, BAF receptor, they can really reach better effects than with 4-1BB. Domain, which is really interesting target for a future optimization and enhancement of the CAR function. Okay, so the last part I wanted from this very molecular part to show you is armoring. Armoring is a huge group of different activities, so I go quite quickly through this. But obviously, what you add on, you can add the suicide gene systems in there. You can add on and off switchers in here. So you can see that suicide system is based on caspases. It could be suicide receptor, a CD20 receptor, truncated one. Or you can have like additional protease domain, and cutting and inhibiting this domain can regulate your CAR. Then you can have, Then what you can have, you can have additional genes that actually express anti-cytokine genes. So we discussed about this EL1 today, blockers. You can do it like within your CAR actually, add it on. Then what you can have, you can disrupt this internal inhibitory immune checkpoints. So this immune checkpoints as a PD-1, again, in the clinics, it's used as an additional drug added to the CAR if needed. Here you can regulate it within your cell, actually. You can obviously add some stimulatory cytokines that gives your CARs boost to expand within the body as well. Then there are some experiments that can mobilize the other cells of the immune system. To express this 4-1BBL, for example, or the chemokine receptors to target the cells towards the tumor. In this case, this helps with the solid tumors. Right. So that's the mechanistic approach about chimeric antigen receptor. What about the cell sources? First of all, we use T cells for now. And we use peripheral blood T cells. And most of the therapies, as you can see in red here, the peripheral blood is a major source of the cells. T cells from peripheral blood are prone to exhaustion, and therefore less differentiated cells and different cell sources are searched and looked for to develop future products, basically. What you can observe is that most of the peripheral blood T cells, they are like effector cells, which can go through several activations and then go into exhaustion, cannot function anymore. Some people try to use stem cells as a source, and these stem cells, however, are on an opposite way. So there are very naive cells in there, and these naive cells means that they lack of cytotoxic activity, so they cannot be good effector. It's quite interesting that several groups already looked for the UCB, so umbilical cord blood, as a source with increased central memory cells, which is like very good would say state for the cells and a subgroup of the T cells that actually can activate and work very efficiently as a CAR therapy. Different cell sources. Again, T cells, when you think about the clinical trials and preclinical ones, T cells are a vast majority. However, NK cells, gamma delta T cells, NK T cells are getting into consideration more and more. So this is kind of an innate immunity cells. So they are not dependent on the TCR receptor, and they can be, although they are less potent in the cytotoxic activity, they have different features that would be good for use depending on your application. These all cells differ between each other, but what is a clue and what is the crucial part is that they all express like cytotoxic activity, because these cells, this is something that you need to remember, these cells are your effector cells. Without them, you don't have this therapy working. And interferon gamma production as well. Right. So what are the hurdles of the current clinical CAR T therapy use? First of all, it was mentioned by Professor Gill before that there is a low quality of the starting material. Low quality of the starting material, so meaning apheresis from a heavily treated patient, gives you low quality of your product, basically, afterwards. Then this increases your manufacturing time and costs. Other parts that are really tricky right now, this is trafficking and recognition of the tumor cells. So you cannot go into the solid tumors directly. You need to enhance this somehow. Tumor microenvironment in solid tumors is really harmful. I mean, what is observed is very, most of the CARs just stop in there and cannot function because it's very inhibitory. Then we have this on-target of tumor toxicity. What it is, actually, this means that actually even if you are targeting CD19, for example, classical, you are targeting all the B cells. We are not targeting only the malignant B cells. This means the question here and the good thing is that basically B cells, we can live without them. Yeah, that's why more or less we can somehow enhance this clinically, but we can live without them. And this is why, like patients after B cell treatment, so CD19 CAR therapy treatment, they basically do not have B cells. Yeah, this is more tricky when you go into the more solid tumors or other targets. If you want to target like a specific marker gene for the kidney, you cannot just destroy all the kidney, right? That's why you need to really better control your CAR, make it more specific in a way, and optimize this one. And this is something that we need to really work on. But what are the perspectives of this therapy? First of all, we are right now at the age of the autologous therapies. So allogenic therapies, surely, yes. Many people are working on that, and I will show you in a moment, to get it like a higher quality starting material and lower cost of production. So to spread this therapy among people and the population. This is quite tricky at some point to engineer these cells, but I will mention it a bit later on. There are solid tumor therapies. So 90% of cancers are solid tumors. This is the way where we need to go. We need to test the CAR-Ts to get into that. Repurposing of the therapies. So there is an example that the CD19, or obviously the CD19 CAR therapy was used in autoimmune disorders. I think that was lupus or something. So you can use this one to control different diseases. Obviously, there are new therapeutic areas, and from this year, this is a quite nice science paper that actually CAR T cells can be used for in vivo treatment of the cardiac injury. So you have a fibrosis, and you can control the fibrosis using CAR Ts that directed. That's the quite interesting idea as well. And obviously, the ultimate modification. So kind of a uniCAR, supra-CAR type, meaning like you put the population of your CAR cells without direct antigen binding domain in your body, and after something happens, you have like disease anti-CD19, you have disease that you need something else, then you just add it on, just this motif, and then you can really activate your cells within your body to target the specific disease you have. And this is something I believe we will go towards. However, this will take a time for sure. Okay. So going back to the stage, going back to the ground, would say, from all these mechanistic approaches, let's go through the current clinical state of the art. As we've seen on the last lecture, this is CD19 and anti-BCMA CARs that are applied in the market. I will focus just on CD19 for now, because they are targeting B cells. What we have in here On the market, this is these therapies, Yescarta, Kymriah, and Breyanzi. So they do not differ from the molecular point of view much. They have the same signaling domain, and just a bit differences between this between the signaling domains and the transmembrane, but the antibody is exactly the same. What is the target for them? Starting from the large point of view, this is 18 million new cases of the cancer, every type of cancer worldwide, right? Then we go to the more kind of hematological cancers, which are around 10%, 6 and 5. And then NHL, so new non-Hodgkin lymphomas are 500,000 cases yearly. This is 3% of cases of cancer. Then DLBCL, which is one of the most investigated parts and most common targets for the CD19 therapy. And these are around 34% of NHL cases. This means that actually in the third line of the therapy, we have in US and Europe only around 10,000 people. Right now, Professor Gill already showed us that there are like second line therapy in the LBCL as well applied, and this increases it to 25,000 people. And what was expected in 2018, this is a patient's report that they said, okay, this means that if Kymriah therapy is already on the market, so that means that yearly we will add on like 15 to 20,000 of the patients, and until 2027 we will reach almost 200,000 patients treated by CAR-Ts. This is what they expected. This means that actually today we should have around 70 to 80,000 patients treated. And we've seen this from Professor Gill already lecture, that we have around 3,000 would say in Europe patients treated till now. So it's 5% of all. Maybe 10% if you add all other cases in the world. 10% of what is expected. And the issue here is not the clinicists. You've heard clinicists how they feel about CAR-Ts. They want to put it. There's a different issue, and the issue is more biased by us. This is manufacturing, basically. We cannot make it. It's too much. Nevertheless, there is an intensive clinical trial landscape. You can see that from 2016, a lot of trials happening, and it's still increasing. We are transferring towards larger phases, so this second phases and the third phases, they are appearing. So it means that new products, that will be never a type like that we have only one antibody and one producer that is gaining all the market. Right now, that's surely because this is autologous cell therapy, so it means that actually there would be a plenty of the manufacturers that need to supply the patients individually. That's the difference from where we are changing from. I mean, from the chemically based pharmacology towards living drug pharmacology. Then when you need to really make it locally, more or less. Most of these therapies are still developed. There are autologous therapies, allogenic, gaining the field, but not yet there. And most of them are really hematological. So CD19, BCMA, this is all on the market, but the new therapies are coming, and there are like very small, like the rest of them, they are like much less interested in there because it's harder. Especially HER2, this is one of the most popular solid tumor markers, and this is you can see that this is targeted quite well in here. And with the clinical trials, obviously these clinical trials are supported mostly by the companies because like in terms of the academics, although we are really doing that, trying to do that, but after the first phase, it's rarely an academia can support the clinical trial and the development of the product. And you can see that there are quite some, I would say around 90 of the companies that are developing only for the DLBCL. And what is quite interesting, in Poland we have one as well. In Poland we have Famicort Therapeutics, which is developing these type of the therapies, and which I'm happy to be part of as well. And this is a spin-off biotech company that is working on this implementation of these CAR therapies. It was founded 3 years ago, actually started working 2 years ago, very intensively on the CD19 product as well for DLBCL. And right now we are at the stage at the just before starting the first clinical trial for that one. This is the pipeline for the Pharmacort therapy, Pharmacort therapeutics. First of all, it's like, again, this hardware as everyone has. So we have the same tarcidome chimleucel. This is like anti-CD19 therapy that we would like to go with the CD19 malignancies. However, we would like to go as well with the non-canonical treatments. We have some allogenic approach, solid tumors, new targets. And we have quite good manufacturing developed already. So tarcidome chimleucels, you've seen it before, so just shortly, this is Pharmacort TX differs, it's quite similar to the Yescarta, Kymriah, Breyanzi, but actually what we have a new molecule that is binding the antigen. So we believe, based on our preliminary results, that this could be a bit changer on the market. I will not go into details what we needed to do, how much we needed to focus on the manufacturing, on the scaling, on the optimization of the cells to get into GMP. That's the 2 years work of 15, 20 people, and it's quite a lot to be done. What we can have, and already done, actually, what we can say is that the phenotypic of characteristic of our product, based on optimized conditions, we have more helper cells, so it means like together with this 4-1BB, as I mentioned before, we expect our product to be more prolonged during the longer time. So the appearance of this product should be seen in the patients for a longer time. That means that hopefully overall survival rate would be better, and this is something that we expect. Obviously, we have great results in the mice model showing this the tumor is not there. And right now, yeah, as I mentioned, we're just before starting the first patient. With the allogeneic approach, so with most of the allogeneic approaches, they are targeted. Like from the scientific point of view, what I would do, I would like engineer the cell to get rid of these all MHCs, TCRs, right, to get it more allogenic so I can put into all patients. And this is great from the scientific point of view. However, from the manufacturing point of view, from a clinical point of view, more gene disruptions you apply there, it's worse, right? So in these terms, when you think about, okay, I would like to have induced pluripotent stem cells, that's the ideal one, which I love and I work a lot with, and I have even a group of advanced tissue models that are based on the iPS cells, but in this particular way, we need to induce the pluripotency, so make this gene disruption, then we need to go through the viruses, which is then gene disruption, then we need to do as talents or CRISPRs or whatever else, still again, gene disruption. Then you select one clone, one cell, and expand it a lot for thousands and thousands copies of the product. This means that you are really making these cells prone to mutations. And this is something that you need to keep in mind, and it's already seen when people are developing these therapies, and FDA is holding on their trials because there's something happening there. What the idea is, based on our infrastructure we have, is that we'll go through the banks, allogenic banks, ready to be done, ready to be used off-the-shelf therapies with the modified CARs, like adjusted to the population of the, like, for example, for Poland, right, for a different country. This means that actually if you're going to get the CAR T cell therapy, your therapy is already there, matching your HLA. This is very good and very interesting because they are immunologically permissive. Cord blood cells, T cells, are less mutation prone because they are younger, basically, so they are having less somatic mutations as well. And we do have armored CAR construct under, right now in our terms that can enhance their function. So this is really good, and we are not the only ones, believe me. MD Anderson is one of the examples. They are really hardly working on the cord blood cells right now to make a CAR. we would like to cope on that one. Basically, what this gives us, this is less risk, more safer mutation, less mutation, safer therapy. And this is a CAR-T inclusive project that we are right now developing. The last part I would like to discuss with you is about manufacturing. Most of the biotech companies, what they do, they are started by the scientists. So I develop my technology and then go, let's try put it in the clinical trial. We had this chance to have infrastructure ready, and we've started from a very scratch to develop our own platform, to not be dependent. So we develop an independent manufacturing platform so that we can design and we can develop and produce every car you can imagine from the scratch. This creates an independent manufacturing organization, as I mentioned, and this is quite easy to scale up and spread. We have these 3 vector manufacturing, CAR-T manufacturing, and quality control in-house already. And this could be spread easily through the group GMP labs that are across Europe. So scaling up, it's quite easy. The process for the manufacturing, it's not so easy as we imagine from our side. From here in our labs, as we can do CAR-Ts, we can do it like within a week. But here it's much, much more complicated, I would say. So the quality control assays that you need to do during the process, there are over 60 of them. And each of this quality control process need to be validated. And this validation means that you need to have from 3,000 to 5,000 reactions for each to make it validated. So it's 60 times. And this was quite complicated process to go through it. But yeah, generally the process for the scientist is quite easy. You can go and expand the plasmids here. Then from the plasmids you produce an lentiviral vector, and then from the lentiviral vector you put it on, transduce the cells, and get the CAR-Ts. From these 3 major parts, I would not go through it, but I would just show you that there are plenty of analysis. Obviously, you need to check the sequence, you need to check the way how the plasmid behaves. You need to check how pure is your vector here, because it's already part 2 with the vector, and how stable it is in different conditions. And with your CAR-T product at the end, that's just examples of the analysis. You need to even check if your products taken from the cryo bag against the same, how much of the product you can take from a cryo bag, and this need to be validated as well. So it's very complicated process to get into the clinics. However, we believe we are almost there, and yeah, that's basically summary I wanted to tell you. And I want to thank you for listening this talk, and please remember that evolution always needs to start somewhere to get into some place. And I would like to thank 2 groups actually that I have the pleasure to work on with. This is human genetics head molecular pathology department actually, that I can really develop my love to science, and the Pharmacothetics group, which is which gives me my strive to put products into patients, and obviously great people there as well. And I have to thank both very nice leaders I have, both Natalia and Tomasz. They are like people who actually always sees opportunities when something happens, not rather risks and malfunctions. So thanks to them, this can really catch on and it works. Thank you very much.
Thank you, Tomek, for this really comprehensive presentation. Are there any questions for Tomek? If not, maybe I will start. So you know that I am also the IPS fan. What do you think about the haplo-identical IPS cell bank as a source for CAR-T?
Yeah, that's obviously that's a point. But this means actually, so that's a great idea, and I'm even part of the cost based on that. So to create a haploidentical iPS cells bank for populations in Europe. However, here it's another story. So one thing, you don't need to modify it. So you just produce thousands of products, right? You make the bank from the iPS cells. Now the other story is that the iPS cells are still not in the clinics, right? So you can differentiate, and this is really great job done a few years ago, among the last few years actually, that the iPS can be differentiated into T cells, into preliminary results. I would say these T cells are really naive. So the function, the cytotoxic function of the T cells from the iPS cells, it's something that's still it's need to some work on. The second thing is that what about the iPS? iPS already have the mutations, somatic mutations, because you get it from the somatic normally cells. So you have already some mutations in the iPS in the cells already, and you need to expand them anyway. So that's something. I mean, you would like to go to the as naive possible population, which was not which was not open to somatic mutations, basically. Yeah. So that's my feeling. It's something you need to take into consideration these facts.
I have the same feeling that there is still a lot of validation and safety concerns about the iPSC therapies, but I'm sure that we still have to work on it because this is the future. Are there any other questions?
Great speech, great introduction. I'm happy to see that you are so close to the final stage to get to the clinic, and I really enjoy your story about cars here at the end, especially that it's not about developing of a car, but some cars were going left and some right. So you might develop, but still going in the wrong direction. So my question is just very theoretic. How do you think you are going to the patient-specific car or universal for everyone?
Yeah. Sorry. Yeah, great question. So I believe that we need to go through this patient-specific cars. That's the stage that we need to. That is the lesson that we need to really have. So you need to go through this autologous car. We started with autologous CAR, that's the one thing. We right now going into allogeneic, but doesn't mean that allogeneic would be better. Allogeneic would be cheaper. So it means it would be applied maybe for much in a cheaper way. I mean, because you can have a bunch and just put it on to the stocks. But this doesn't mean that actually they would be better for single patient. That's always something that we need to think about. Because the question was about should we go with the specific car anti-something, right? Anti-target, or that would be unicar more or the supra car modified. Yeah, well, in these conditions, in these clinical conditions, surely we need to go and check for the possibilities that we have with the specific cars. That's something we need to do, because going with the unicars this is very fancy and very nice. I like the idea, basically. But then you add some molecular aspects that you do not consider. For example, I've just mentioned about the steric issues, right? So sometimes you need to CAR, which is like a longer one. It's constructed in a different way to reach the target. Sometimes you need to target the cells. So your CARs shouldn't be CAR T. They should be CAR NK, for example, or CAR macrophages CARs, so M CARs, for example, right? Because you would like to target the environment where macrophages are working better. So it doesn't mean that only the T cells are target cells. I would like to see the unicars, how they develop, but I believe in clinical use, that would really take a time. Yeah, that would really take a time.
Perhaps more even psychological question or philosophical question. Yes, because it's really, the technology is really incredible, and even more incredible is that we are using this in hospitals, in our hospitals, with all these different instances. And I'm just wondering, of course, why we use it, because it appeared very efficient in patients that have nothing to lose. And this was probably something that helped us to break this emotional barrier of using genetically modified cells. But what do you think? Where is the limit of genetic modifications in humans? Yes, because we can introduce more genes, use CRISPR-Cas and differentiate iPS cells. Think about everything, but well, I'm just thinking whether we can do whatever we want right now. Perhaps more closer to the ground question about the safety precautions when you are developing new cars or new car constructs. What are the practical aspects that should be taken into account so that the cars can be finally given to the patient?
Yeah, thanks. It's a hard question, really. I mean, where we should go. I mean, but keep in mind, when I'm thinking about this, I mean, I always keep in mind that, we needed such a trigger, as you mentioned. I mean, there was like no option patients, and we put in there like gene-modified cells, so genetic gene therapy, and it worked well. This really opened. We are in a different era right now, I would say, than in 2010 or 15. This is different era. People are really accepting this for now. You can see EMA and FDA accepting the second stage already, right? So second line therapies as a CAR-Ts. So it means it's no longer an option group. Actually, we are right now in the optional group, but it's better. Yeah. So what about safety? Yeah. That's the major precaution. That's why that's why it's not that every academic lab can do this therapy, right? Because it's so many things to be controlled, so many things work to be done. So think about like there are huge companies. There are like huge companies, really, like Gilead, like Novartis. There are like big ones, and they wanted to put this therapy on the market, right? There are so many people that want to have this therapy and need to have this therapy, and they cannot do it. It's not so easy. And they are really doing like small drops right now, 10% of the expectants. So so this is something that, it's good. And basically it's good. It's developing side by side, step by step. So to ensure that therapy is safe. Where we should go with these iPS cells, for example, I believe that iPS cells will be used in the clinic, and I'm pretty sure of that. I'm not sure if in every use, like the same for the CAR-Ts. They are like for specific. Maybe we can do the solid tumors with the CAR-Ts, maybe not. Maybe there is a different technology needed to be developed. The same is for the iPS cells. I mean, there surely are something for the future, but not always, and not in every direction they should be developed, right? In this particular way, as I mentioned to you, I believe this is already seen. I mean, in the clinics, we have like 100 patients treated in a second phase trial, and there are something going on. Obviously, okay, FDA said this is, there are like some tumorigenicity in these patients that were given these allogenic CAR, modified CARs. They said that they didn't detect that there was through the CARs, but still we are only under the 100 patients. What about when we add it to the 3,000, right? I mean, how this will look like. It's tricky. Safety first, that's for sure.
Thank you.
Okay, so we have two more questions from chat. The first one is, there's a resounding name of trucks for some type of fourth generation CAR-T. Are different than trucks fourth generation CAR-T really used?
So the trucks, thank you for the question. Basically, fourth generation car, I didn't show it up on the picture, but it was on the slide. Fourth generation cars, these are cars with this armoring. So enhanced cars somehow. Which is quite interesting because third generation car is not really often used, but the armored cars, so this fourth generation, they're really in a preclinical stage, but very intensively developed. Trucks, so car, you've seen how the car look like, the car construct, the chimeric antigen receptor. Trucks are more constructs which are basing on the TCR. So they are closer to the TCR biology and function. The issue there, I mean, the idea there, it was like, okay, if we are closer to TCR, this would be better controlled because TCR has different switchers and all the complex need to be handled somehow, and this is better control, and it is like that would be safer therapy. However, it seems that it's too complicated clinically somehow. At least this is my opinion. Maybe that will be different, but from my side, it's like sometimes in the clinic we need to go. I mean, very often in the clinic we need to go simple. This is not the scientific idea. It's how to make it working. Sometimes simple is better.
Yeah, I agree. Sometimes in the paper it looks really fancy and complicated, but when it comes to the clinic, it's the simplest safety is the best solution. Okay, then the last question. Are we anywhere near 5th generation, or we just focus on improving the 4th generation CAR-T?
Yeah, I think at some point people just stop naming the generations at some point. So yeah, and this is what we have right now. We develop obviously this armoring in a more fancy way, in a more delicate way. These armorings are a lot of them. I don't see the place. I mean, in a way, okay. At some point, we are reaching the capacity of the vectors, capacity of the transfer genes, and we cannot just put as much as we can genes inside. So that's about safety, as was mentioned as well. Yeah, so at the end, 5th generation is just a name. Yeah.
Okay. So, are there any more questions? If not, thank you again, Tomek, for this really nice presentation and the discussion. And I would like to invite you to the coffee break and be there at 1:30 because we will start the last but not least exciting lecture given by Professor Basak. So see you in a half hour.